Namaskar. Today we will study small interfering RNA, siRNA, which is a non-coding RNA. And non-coding RNA, we can have two types of RNA, coding RNA and non-coding RNA. Messenger RNA is the coding RNA and the siRNA is the non-coding RNA. And non-coding RNA are derived from the intron part, whereas the coding RNA are derived from the exon part. And this non-coding RNA can be housekeeping or regulatory and can be long, lengthwise more than 200 nucleotides or small, less than 200 nucleotides. And siRNA is a type of small non-coding RNA. And this siRNA will have about 21 nucleotides, and this is exogenous. Unlike our microRNA, which is endogenous, this siRNA is exogenous. It can be delivered through viral infections, other infections, or through the transposable elements. They can be transferred, and usually this is transferred in the form of the double-stranded RNA. Then it is inserted into the cell, so it is available in the cytoplasm. And as we know, in the cytoplasm we have one enzyme, one RNase 3 type family enzyme called dicer, which will bind to this double-stranded RNA and will cut it into small 21 to 23 nucleotide molecules called small interfering RNA, which will then interact with another endonuclease enzyme that is Argonaute, and basically Argonaute 2, and form this complex called RISC, RNA-induced silencing complex, which this siRNA is having two strands. One is the guide strand or antisense strand, and the other is the sense strand or anti-guide strand. And the sense strand or anti-guide strand is degraded, where ATP will be required to provide the energy, and the guide strand or the antisense strand is now required to silence the post-transcription gene expression. So, say the messenger RNA, this siRNA will now bind to the messenger RNA. But in case of microRNA, there is incomplete or partial base pairing, whereas in case of siRNA there is complete base pairing. So there will be mRNA degradation. So this is the major event in case of siRNA. mRNA degradation will be there. So if there will be no mRNA, so no protein synthesis will be there. And in case of incomplete base pairing, which mostly occurs in case of microRNA, and it can also occur in siRNA. So there will be ribosomal block, and there will be stoppage of translation. So there will be no translation. And sometimes that means this is one way, and the other thing is this guide strand complex can enter into the nucleus, and can bind to the target DNA and can form a complex called RITS. Here RITS stands for RNA-induced transcriptional suppression, T for transcription. So RNA-induced transcriptional suppression. How? By causing this histone methylation. So there will be methylation of the histone proteins, and there will be remodeling of the chromatin. So it will result in the heterochromatin formation. As you know, if there will be heterochromatin, there will be no transcription. For transcription, the U chromatin is required, and in case of heterochromatin, there will be no transcription. Then there will be transcription repression. So small interfering RNA does mainly one mRNA degradation, and number two transcription repression. So we can see in any eukaryotic cell there are 6 targets where the gene regulation can be there, and siRNA mainly targets this target 1, where this euchromatin will be further condensed to heterochromatin because of the proteins activated by siRNA, and there will be no transcription. And second thing, this fourth step where mRNA degradation occurs, and this translation may occur, but this is to a low extent. So these are the ways in which small interfering RNA interfere the messenger RNA action, post-transcriptional action, and cause gene silencing or can make the gene off. And so we can transfer this siRNA as a drug using one artificial vector, and it will reach the target genome and bind to the target genome or DNA, and there will be target specific gene silencing. So siRNA is nowadays under investigation for use as gene therapy, mainly in case of cancer, infectious diseases like hepatitis, HIV or AIDS, and nowadays also they are used for neurodegenerative diseases like Alzheimer's disease, Parkinson's disease, and we can use them as insecticide or pesticide by targeting the insects and pests to silence crucial genes in the insects and pests. And we can also use siRNA for production of plants with lower level of natural plant toxins. For example, cottonseed, the toxic substance is gossypol. We can lower the level of gossypol in the cottonseed by using specific siRNA. So these are the uses of siRNA. Thank you.